Welcome back. Now we have two more talks in this session. The first talk is by Rajat from NCBS, and who's going to talk about RNA and condensate's relation with transcription factors. So, over to you.
Thank you for the. Hello, am I audible? Yeah. First of all, I would like to thank the organizer for inviting me here. I am Rajat from Dr. Dimple Notani's lab at NCBS, and I'm going to talk about how RNA provides specificity to condensate and transcription factor binding. So my work would be majorly focused around these proteins called transcription factors, so that recognize and bind to certain sequences in the DNA. So what it does if the sequence is exposed, by meaning exposed if it is not embedded inside the nucleosome, then transcription factors are proteins that are known to bind to these sequences, and they recruit different activators like P300, mediators, and as well as the polymerase machinery to the site, which subsequently lead to the transcription of the said gene. Now, what I wanted to understand is how does the binding of what are the different factors that regulate the binding of transcription factors? So in order for the cell to regulate the transcription inside the cell, it is important for the cell to regulate the binding of these transcription factor. So first is the motif itself. So transcription factor doesn't bind to a single motif, it binds to a consensus of different motifs at variable strength. So the strength of the motif is how similar that particular region is to the consensus motif. The higher the similarity, the higher is the strength, and the higher is the binding of the transcription factor. So in addition to this sequence, the DNA information contained inside the DNA, there are other factors which can also regulate the binding of transcription factor, which would be the focus of my talk. So two of these factors are, first, the transcription. How does the process of transcription in turn regulate the binding of the transcription factor? And the other is, how does the output, that is the RNA itself, how does that regulate in controlling the binding? So the model system that we have used is an estrogen signaling system. Estrogen is a steroid hormone which can translocate from the extracellular milieu to the inside of the cell, where in the cytoplasm it recognizes and binds to the receptor called ER alpha, which is the transcription factor in question that we are going to talk about. And upon binding to the receptor dimerizes and it translocates inside the nucleus, where it binds to its own motif called the ERE, or the estrogen response element, and which, like other transcription factors, it would lead to the transcription of these genes. Now what I wanted to understand is how transcription as a process feedbacks into this. So transcription is a multi-step process that requires initiation, then followed by elongation, and then leading to termination. So what I wanted to understand is, if you perturb transcription, how does the binding of the transcription factor gets perturbed? So we use this particular drug called DRB, which is a elongation inhibitor. So what DRB does is it would prevent the transition from the initiation to the elongation phase. So the initiation rates are the same, and that would lead to the increased accumulation of polymerase to these sites. So which, after treating the cells with these DRB, there would be an increased amount of polymerase to these sites. Now that, along with E2, so first I treat the cells with DRB, which is the inhibitor, then to stimulate the binding of the transcription factor, after treating the cells with DRB, I add E2 to the mix. And after 1 hour of E2, so every E2 signaling system has a peak, and that peaks come as around 1 hour. So at 1 hour, majority of the transcription factor is bound to the DNA. Now what I wanted to see, if you perturb the transcription using this drug by increasing the polymerase at these sites, how does the binding of the transcription factor gets perturbed? So to assay the binding of transcription factor, we employed this technique called ChIP-seq. So ChIP-seq tells you where exactly in the genome your protein of interest is binding. And what you see here, the end product that you see in these regions are the peaks, or the regions that are the binding sites that are for ER alpha. These sort of triangle-like regions are the binding regions for ER alpha, and this is on one of the responsive genes called GREB1. And what we see here is the binding of ER alpha after treating the cells with DRB is increasing. Now we wanted to see genome-wide, what is the effect of the binding genome-wide. So we looked at all the binding events. We see that there is a gain of around 2,000 peaks and a loss of around 1,500 peaks genome-wide of ER alpha. Now we wanted to see how does the binding profile of these peaks look like. So now we looked at these 6,500 peaks and these 2,000 peaks. So what you see here is a summary plot by summarizing the counts from all of these common sites, the 6,000 sites. And what we see here is that the binding of ER alpha on these regions are going up, which is on the common sites, and as well as there is an increased number of gain sites as well. So combining both of these, the increased binding on the common site as well as an increased number of gain sites suggests that there is an increased binding of the transcription factor genome-wide. Now what we wanted to understand is what regulates the binding of the transcription factor now, after treating the cells with these inhibitors. So we already knew that motif is one of the things. The information contained within the DNA is one of the feature. So first we looked at the regions. So these are all ER alpha peaks, and all the ER alpha peaks are binned on the basis of the motif strength. So high motif strength would mean that it is more similar to this ER alpha ERE motif, and low meaning it is less similar. And within each bin, the motif, the underlying information is the same, but one thing that is varying is the polymerase levels. So 1, 2, 3, 4 means the increasing level of the polymerase. Now we wanted to see if we normalize one variable, that is the DNA information contained within the DNA, how does the binding of the transcription factor after you treat the cells with DRB, how does that relate to the polymerase level now? What we see in the same bins so this is log to fold change values for DRB over DMS row, quantifying the increase. And what we see here, the regions that showed the highest pol2 were the regions that were showing the most gain. So wherever there is a higher increase in the polymerase, those are the regions that are showing a higher increase in the ER alpha binding. So to summarize, what we have seen here is in a normal situation, transcription factor would recruit the polymerase, and that would lead to the transcription of the cell. Now, after you treat the cells with these inhibitors, what would happen is there would be an increase of the polymerase because of the drug, and now which would lead to increase in the transcription factor binding. And where exactly transcription factors are binding, these are the regions where already had a higher polymerase level. Now the ChIP-seq experiment tells you a population, like if you take millions of cells, what is the output from the millions of cells. Now what we wanted to see what actually happens at a single molecule level. So we use this technique called single molecule tracking experiments. So what you do in this, you take the protein of your interest, you label it with the given tag like halo tag and that, so that you can vary the dye concentration so as to get a very less number of particles labeled, and from that very less number of particle label you can get the trajectories of those particles, and from those trajectories you can get a rough idea of how fast they are moving, how if they are diffusive, if they are moving within a confined population, or other factors. And other thing what you can also calculate is the residence time. The residence time tells you how long a given transcription factor stays at a given region. So we did the same thing with ER alpha. This is ER alpha labeled with halo. We did it along with Gunjan from IIT Hyderabad. And the small blips that you see are the single particles of ER alpha. And we did these two experiments in both DRB and DMS to quantify how does the motion of these particles change after you treat the cells. So what we find here, so unbound fraction is the diffusive fraction, the one that is moving freely, and the bound fraction is the one that is subdiffusive. It's not moving freely, but it is something is perturbing the binding of the transcription factor from moving it freely. So what we find is that in the case of the unbound fraction, we see that there is a decrease in the unbound fraction, and there is simultaneous increase in the bound fraction. This is similar to what we see with ChIP-seq as well, the population level, that at the single particle level, we see that the bound fraction of ER alpha that is bound to the DNA is increasing. Now we just wanted to see what exactly is happening with the bound fraction, the protein that is actually bound to the DNA. So we performed the same imaging experiments at 200 milliseconds, because at 15 milliseconds what you do is you keep the laser power so high to get the particles, but what can happen is these particles can get bleached. So to get a better idea of the transcription factor motion, so we did it at a lower frame rate of 200 milliseconds. And what we did is we calculated the dwell time. And from the dwell time, we did it in both the DMS as well as in DRB condition. And from the dwell time, we get a survival plot of how long a given particle is staying at a given time. And we see that majority of the particles are staying at a given location for a lesser time, but as the time increases, there are few particles. And we fit this into two different populations, a bi-exponential fit, and what we get is two different populations. First is the population that shows the dwell time of around 2 seconds, and the other is the population that shows the dwell time of around 10 seconds. The population that shows the dwell time of 2 seconds is the transiently bound fraction, which is bound to the DNA for a lesser time, and what is bound to the DNA for a longer time, that is of 10 seconds, we call that as the stably bound fraction. So after treating the cells with DRB, what we find is that the dwell time of the transcription factor went from 10 in DMSO to 13, that is in DRB, and For the transiently bound fraction as well, it went from 1.9 to 2.906. It's a very, very subtle change that we see. To summarize, what we have found here is that as you inhibit the elongation using DRB, what we see is the polymerase gets stabilized, and because of the stabilizing, we see an increase in the chromatin bound fraction. And once we calculate the dwell time for the CR alpha, we also see that there is an increase of the dwell time, which is sort of similar to what we see for what we saw for ChIP-seq experiments as well. Now, ER alpha is known to form these condensates. People have reported, us and others in the field have reported that, so this is ER alpha inside the MCF7 nucleus. And as soon as you add the ligand, we see that there is a formation of these condensates here. And now what we want, so now all of a sudden why I'm talking about these condensates is because people have talked about that there are certain these condensates, especially for MED1, that is selectively is known to partition these positive regulators of transcription like RNA polymerase and other things, and which are in turn, people have shown that it is sort of related, showed a positive correlation with transcription. And another factor is that we us have previously shown that these condensates have a genomic basis. So what we had done in this study is that we have deleted one of the regions, and now we checked, does the condensate formation on those regions change? And we saw that after deleting the region that is known to bind to ER alpha, the formation on the condensate on those regions change. So we think that these condensates sort of have a genomic background. So wherever there was a higher binding, those regions, multiple of those regions are coming together to form these condensates here. And from the previous experiments, we saw that genome-wide, the binding of the transcription factors was increasing, as well as how long a given transcription factor stays at a given location, that was also increasing. Now we wanted to see how does the condensate dynamics change in these. So we labeled ER alpha with EGFP, and we followed it with time. So the time that you see is time post addition of the hormone. Initially, it's a homogeneous, the pattern is homogeneous, and after you add the ligand, we see the formation of these spots. We quantified the formation of these spots by two different ways. First is we calculated the number density. How does the number of these things increase with time? So we see that initially, since the signal was homogeneous, we didn't see many number of these condensates, but as soon as the time increases, we see that it sort of reaches a plateau phase. So the other thing that we calculated is the size of these condensates. So this is the average diameter of these condensates, and we see that also sort of follows an exponential increase, followed by a plateau phase here. We did these two experiments after treating the cells with DRB, and what we calculated is first the number density. We see that we calculated two different parameters. First is the maxima of the curve, that is what is the maxima of both the curves here. And what we see is the maxima in terms of the number density is not changing much, but we wanted to see how fast are these condensate forming. So we calculated what is called as the t-half of these curves. So how we calculate t-half is by fitting an exponential curve in these, and from that we get an idea of the rate at which these things are increasing. And t-half is the measure of how long it takes for it to reach half of the maximal response, and we did it in both these conditions. And what we see here, in the case of DRB, we see that the formation of these condensates are faster, as it is sort of evident here as well. Next, we looked at how the size of these condensates are increasing. We see that in the case of DRB, we see that the formation of these condensates are bigger. If you look at the max here, the max curve between the two, the DRB and DMS, so is quite different, and which is the same thing quantified here, that the max diameter between the DRB condensates, the ER alpha condensates in DRB and DMS is different. But once we looked at the diameter T-half, that how fast the diameter is increasing, that didn't show us drastic differences. So now what exactly is so with the confocal live cell imaging, what we get a rough idea of two different populations. First population is one that is freely floating inside the nucleoplasm, and the other that is actually bound to the DNA. So what these increase with respect to the T-half and the increase with respect to the size can be attributed to both the population, if it is because of the freely floating protein or it is because of the ones that is actually bound. Now, since we were interested in only the protein that is actually bound to the DNA, so we treated these cells with this buffer called CSK, which is known to remove the freely floating population from it. So, and we performed immunostainings for ER alpha after treating the cells with this buffer, and just to get just the DNA bound fraction, we calculated these ER alpha condensates volume fraction, and what we find here that in the case once we look at the volume fraction in the case of DRB, which is also showing a similar increase, which was showing in the case of the live cell imaging. So to summarize what we have found here is that in the case of DRB, since the dwell time which we saw from the single particle tracking experiments was increasing, which can lead to an increase in accumulation of the transcription factor, as well as other factors as well. The polymerase might also be present in these regions, and which lead to a subsequent increase in the formation of these hubs. So now what we wanted to understand is how polymerase is actually doing it. What is the mechanism? How does the polymerase lead? Yeah.
Sorry, I have a question.
Yeah.
You've drawn this as a condensate, so are you saying that a single condensate is just one single complex?
No, there would be multiple. It's just
Okay, it's
just
just
a representation.
Okay.
I'm not saying that it's a single complex. There can be multiple of DNAs coming together. So, coming back to what I was talking. So, what I wanted to understand is how polymerase is doing these things. So, there are two different mechanisms how polymerase can assist the binding of transcription factor. First is, like I told you before, that the transcription factor cannot bind to the motif if it is embedded within the nucleosome. So for it to bind to the motif, it needs to be exposed. So for that, something has to evict out the nucleosome to free the binding site for the transcription factor. And polymerase is one of those factors which is known to recruit remodelers and other chromatin remodelers to these regions, which can lead to eviction of the nucleosome and which can expose the binding site for the transcription factor, leading to the stabilization that we see in the case of transcription factor. The other thing that can happen is the RNA. The RNA, so many of the transcription factor, beside having a very well characterized DNA binding domain, also have a less well characterized RNA binding regions. One of the less well characterized regions is already talked about before, the RRGG motif here. So the transcription factors like having those RNA binding regions can in turn be present in the proximity of these the motifs, and which can lead to the feedback to these motif regions, which can stabilize the binding of the transcription factor. Now, it is already known that once you treat the cells with these inhibitor, that is the flavopiridol is another elongation inhibitor, people have shown that as the polymerase increases, so does the binding of the nucleosome remodelers also. And that in turn can also lead to the opening up of the chromatin, like I was talking about. And now this can lead to exposing the sites of the transcription factors or the motif, which can lead to the stabilization. But for the case of the transcription factor, so ER alpha, like other transcription factors, also have a less well characterized RNA binding domain. And this is an RNA immunoprecipitation study which we have done here that is known to interact with TFF1 eRNA, which is another eRNA present in the nucleus. And this interaction sort of increases once you treat the cells with E2. So ER alpha is another transcription factor which can bind to the RNA, but the question still remains that now I am treating the cells with the inhibitor, so there shouldn't be any RNA because the elongation step is getting affected. So there shouldn't be any RNA coming out of it. So, but that is not entirely true. So there is a population called the short RNA that comes out. So this is the RNA that is shown here. So after you treat the cells with these inhibitors, what people have shown that there is an increase of this short RNA that comes because of the polymerase getting stalled. The polymerase secretes this short RNA because of not proper elongation. So this short RNA can in turn affect the binding of the transcription factor. So now what we wanted to see is we wanted to decouple the effect of the two. So we took a stepwise approach. First, we treated the cells with this another inhibitor called triptolide. Triptolide would degrade the polymerase and which would also lead to a decrease in the levels of the RNA as well. So both RNA is not there and the transcription factor is not there. Now we assayed the binding of the transcription factor using ChIP-seq, and what we find here is the binding of the transcription factor goes low. So this is the same thing. So these are ER alpha peaks and the counts is shown here. So the higher the blue means the more the loss in these, the higher red would mean there is a gain. And we see that after treating the cells with TRP, that is the triptolide inhibitor which perturbs this, we see that the binding of the transcription factor goes low. So in the condition when there is no RNA, no polymerase to open the thing, we see that there is a decrease in the binding. Now what we wanted to see, what would happen if you were to increase the level of RNA without affecting transcription. So how you do that is there is a protein that is known to degrade the chromatin associated RNA. So this is called the exo-SC3, which is part of the exosome complex. So if you were to knock it down, then the levels of the chromatin associated RNA goes up without affecting the process of transcription. So now what we have in this system is that the levels of the chromatin associated RNA is high without the transcription being affected, and now we checked at the binding of the transcription factor. We see that after we knock down this, we see that the binding of the transcription factor goes up, and which is also shown at the genome-wide level as well. Now we know that the transcription factor, the RNA, is sort of supporting the binding of transcription factor. Now what we wanted to do is decouple the effect of two. If it is the RNA or the transcription factor or the openness, how does that work? So we took this particular mutant, that is the ER alpha mutant, which is not able to bind to RNA. So in this particular mutant, we have seen that the binding of the transcription factor to the TFF1 eRNA, which I showed before, gets perturbed, and which leads to a genome-wide loss of the transcription factor binding overall. And now what we have is a mutant that is no longer able to bind to the RNA. So in the case if you treat this particular mutant with DRB, now the opening of the particular chromatin would still be there. The polymerase would still get recruited to those regions, and which still gets stalled near those regions. But what would happen is the openness would still be there, but the RNA which is coming out of the stalled polymerase would not be able to interact to this ER alpha, because the ER alpha lacks the ability to bind to RNA. So now if the polymerase was important, we should still see an increase, but if the RNA was important, we would not see an increase. And this is what we see, that if you were to treat the RNA binding mutant with the help of DRB, we see that in the RNA binding mutant, we don't see that increase which we were seeing for wild type, suggesting that it is the short RNA that comes out of the stalled polymerase that is responsible for these effects. So saying that the short RNA keeps the transcription factor in proximity so as to regulate the binding of the transcription factor to the DNA. Now, which brings us to the question that why exactly cell is doing all of these things. So why is there an increase in the binding of transcription factor? What has cell to gain with these? So I have not shown data for another. So what we have done is we have looked at another global activation mark, which is ST cadence and acetylation, which we have seen that also follows a similar pattern, that wherever ER alpha is increasing, that is also showing an increase in HT cadence and 27 acetylation. Now what we wanted to see, how does that relate to the transcription? So what we did is we sort of allowed the transcription to happen. So this is BRU-seq, so which tells you how the transcription is going on, how the output of the RNA is coming out. So we did it in two different conditions. First is the mock condition, when you have not treated when you have not treated the cells with the inhibitor, and the other is after releasing the cells. So once you treat the cells with the inhibitor, so since DRB is a reversible inhibitor, you can just wash it off and the cells will begin to transcribe. Now what I wanted to see, if the regions that begin to transcribe faster were the regions that had an increase in the binding for these transcription factor, or showed an increase in ST27 acetylation. So one of the things that we see here is one example. We wanted to see if this happens genome-wide. So we bind these entire regions on the basis of how much the increase is after you release it. So 1 to 5 is an increasing order of how much the release, the output is after releasing. So what we see here is the regions that showed the highest RNA after release were the regions that showed highest gain of HT cadence and acetylation, or the ER alpha, the transcription factor, before. So wherever there was a higher increase in the transcription factor or HT cadence and acetylation, those are the regions that showed a higher release of RNA, higher formation of RNA after release. So that brings me to my model, what I'm suggesting. In the normal situation, there is an on and off rate of the transcription factor, so which the transcription factor comes to the regions and then lead to the transcription of the gene. But in situations when there is polymerase pausing, which happens normally as well, but here we are adding the drug to initiate that. And what would happen is, because of the short RNA, that would lead to the stabilization of the transcription factor. And once this pausing is released, that leads to a higher output of the transcription. So for that, I would like to thank my supervisor, Dimple, and my committee members, and the lab members, which are also shown here, and NCBS TFI. Thank you. I'm happy to take questions.
Yeah, please.
Hi, Rajat here. Nice lucid talk. The mutant that does not bind the RNA, how well does it bind the RNA, the polymerase?
So it doesn't bind to the polymerase. It would not. So so in the wild type condition, so you're talking about the direct interaction of the polymerase and the transcription factor. That is what you're.
I assume they bind?
Yeah. So yeah. No. So my bad. So in the normal wild type condition, the transcription factor does bind to the polymerase, which will recruit it. In the RRGG mutant, we have just perturbed the RNA binding. It would still bind to the polymerase to the same extent.
To the same extent?
Yeah, ideally. Yeah.
So, but the model that you're proposing now is that the direct interaction does not matter
Direct interaction for
between the polymerase and the transcription factor. Because you're suggesting that when it doesn't bind RNA, you don't really see the enrichment of the transcription factor, right? So then why does the transcription factor directly interact with the polymerase if it's the RNA mediated binding which is more important?
wide. So because I think all of these interactions are happening simultaneously. So transcription factor is known to recruit polymerase to its site. So wherever there would be a motif, the transcription factor is known to bring polymerase and other things to its site. That is what. And as soon as people have talked about, as soon as there is RNA coming to the mix, the binding of the transcription factor to DNA increases. That people have done it in vitro with just the reconstituted system. And another person from our lab has also done similar sort of experiments. So
I'm just hung up on the fact that why the mechanism is evolved to involve the RNA, because the transcription factor is recruiting the RNA polymerase, right, or it's promoting the binding of the polymerase onto the DNA, right? Now, it's already there in place, so what is the advantage of having the transcription factor bind the RNA?
So in certain situations, what can happen is because of the pausing and other things, so the RNA output might not be that high. So if the RNA becomes very high on these transcribing regions, for other transcription factor, what it can do is the RNA can take away the transcription factors also. So that is another very thin, narrow range where the transcription factors can work, the RNA can work in supporting. So direct interaction and how multiple way interactions can support the transcription factor binding, how it has evolved, I'm not very sure what would be the reason. Maybe because of multiple ways to regulate the binding, that adds another layer. So why it is, but I'm not really sure.
Thank you.
So I'm trying to extract in my mind the LLPS aspect of what is otherwise a very, very fine and well-delivered transcription talk. So in this model here, would I be right in saying that where you show the basal transcription, there's no condensate, where you show the pause release, there's no condensate, and there's a condensate? No, go back to the model.
There is a condensate.
Go back to the model.
Okay.
Yeah, and that where the polymerase pauses, then with the help of transcription factor binding to the short RNA, the condensate is helping to keep the flock together so that it doesn't dissipate, so that once the pause is released, the transcription can resume. So is the role of liquid-liquid phase separation only in that central, polymerase pause thing?
Not really. So even in the situations when there is, like we have not induced the polymerase pausing, we see the formation of these condensates. It
So
would be
because I've heard somewhere that these transcription factories that are, hypothesized to exist, are actually liquid-liquid phase separated regions.
that is what people,
Right? Right? Not just the nucleolus, but many
the other
many
transcription
regions,
factories,
right? Transcription factories,
yes.
where presumably in a condensed state transcription is continuing.
In the condensate, within the condensate, I'm not really sure. People just have done FISH, which is sort of an indirect association with it. Within the condensate, what people have talked about is the smaller the size of the condensates, the higher is the transcription. That is, so if the RNA becomes too high, then that leads to the formation of a shorter condensate.
But it can't get as small as a single complex.
No, not really as a single complex. Then
So how many complexes, and are those complexes paused or transcribing?
So in these terms, people have done.
That's the key question, right?
In
I mean, it's a fantastic talk on transcription, but to understand what is the liquid-liquid phase separation aspect of it, I'm trying to understand.
So here,
Is transcription happening inside the condensate? Or is the condensate stopping the transcription?
No. People have talked about from the, so like one of these studies. Where did it go? Yeah. So here, this particular paper talks about in terms of the how does the binding of the transcription factor. So if there is a formation of these condensates here, how does that relate to transcription? So people have not talked about inside if it is happening. They have plotted correlations that if it is closer to the gene, that makes it more burst. So if the condensate is closer to the gene, that makes it more burst. That is what people have talked about, not a direct correlation there. That if it happens within the condensates or not, that's not. But in vitro experiments people have done for taking condensates and then doing IVT, in vitro transcriptions for GFPs, and those people have done that, have shown that when there is a formation of condensate, there is a higher transcription. That people have done. But in vivo, this is the best that I have seen.
Hi, nice talk. So I'd like to, I have seen like in one of your slides you have shown the increase in the number and size in live cell imaging, right? So is it confocal imaging, like how did you?
So these are confocal images, these are confocal, but we have done super res as well.
Right, because what is the size of the condensates you were
So in these, so these are ensemble averages. So what I have done is I have taken the mask of all these things and combined to get the average size. So it's after thresholding, so after removing these low intensity pixels. So that is done after, but we have done super S with STED and we still see similar things there.
And regarding the number calculation, like you have considered a volume, like how you calculate the number, or you consider single plane and
Yeah. So if it is within this single plane, and then it's just normal thresholding, and then based on, so these are intensity based thresholding, whatever is above that I'm calling those. So and every single frame would have a different, what I'm calling is regional discontinuity. So wherever there would be a blip, that would be called. And post that I'm putting different size thresholds and things. If it is below a certain thing, below the diffraction limit, it would not be called. So those things I'm taking.
Because I'm just wondering, since these are very highly dynamic, they are very mobile, so when you are considering only one single plane and they are going back and forth,
So
is there any possibility of like multiple counting of the same entity?
No. So here what we have multiple counting. So this is at different frames for the same cell.
Okay.
So even if something goes up and comes back again, it would. So what we have seen after a certain time, these things become pretty stable. So if you take it for like a higher frame rate, we see that these are pretty stable. It doesn't move much.
Okay.
So that is what we have seen.
Okay, thank you.
So very nice talk. So in general, these ones that you're calling condensates, it's not a simple kind of a droplet that we see, right? It's a, so what is, in your sense, what is the structure? How do you physically characterize it, or have you thought about that?
So in terms of structure, meaning what are the different proteins that are associated with, along with DNA and those things?
Like a droplet, clearly.
Yeah,
right? I mean, DNA is involved.
Yes, definitely.
So, which provides some sort of a surface.
Yeah.
Now, have you tried characterizing that? Because of course there are ideas that of things like pre-wetting and
yeah, so there is a different protein called FOXA1 that acts as a nucleator for these. So like I was talking about, that the transcription factor cannot bind on its own because it needs something to open it up. So what we have seen is, even before the signaling begins, there are certain regions that are primed by FOXA1, so which is a nucleating factor. So FOXA1 can bind to closed chromatin and open it up. So that can assist the binding of these things. So what we have seen for that FOXA1 kind of primes it. So wherever there is FOXA1, we see the formation of these punctas are faster on those regions. So we are looking in those situations as well. And we have removed FOXA1 from this picture, and we don't see the formation of these condensates. FOXA1, we don't see the formation of these condensates. But that is also within a concentration range. If the concentration is already very high, then we still see formation. So it's within the range.
Yeah. So in general, I mean this, like you are saying that here there is a formation of LLPS. I have seen some study, they said that when there is DNA repair, when there is the cut is happening, then also there is a formation of the LLPS. and there is some other purpose to that. My point is that all these LLPS you are forming, they are recruiting specific constituents, right? How it is even getting regulated? Is that something?
Yeah, so there are a lot of studies that people are doing right now. So one of the ones that I already showed was this Med1 thingy. Yeah, this paper. So they talk about charge patterning. So they talked about that if given charge is present at a given like spacing, so only if the for co-activators, that if it is present, the charge is patterned in such a way for if the charge patterning is similar for similar co-regulators, then it would get partitioned. So in this paper, what they have done is they have taken another different protein and made the charge patterning similar to another co-activator, and now they see that it gets partitioned. So there are people who are working on similar sort of things.
So let's thank Rajat for a wonderful talk.
Thank you.
And let's welcome Viman Jana from IACS for the next talk. I'll give you a heads up at 25 minutes.
Okay, so let me first thank Sandeep and Devan for the invitation, and this has been a really wonderful conference because I got to learn many things which I really didn't know about. So what I'll be doing today is that I'll be talking about one small study we have done in relation, somewhat distant relation to LLPS, and then I'll be talking about something which we are recently doing, is really trying to understand simple IDPs, that how we can think about the landscape of IDPs from a simple theoretical background or theoretical point of view. But let me first start with what I said, the first thing. First thing first, in IACS we work on many different problems, which is mostly, I would say, it's not a biological problem, it's basically a physical chemistry problem. That means we look for problems in biology which can be solved by understanding of physical chemistry or physics per se, and then we try to find out some of the underlying principle what is working there. Mostly that is the theme, and these are the different projects we work on right now. Okay, so first thing I will talk about something we have done in relation to LLPS is that little bit introduction. This you all know by now. So these are very important from different perspectives, but of course the regulation is important. If you get into dysregulated thing, then you get into trouble, having disease and stuff. Like people have done a lot of studies to show that there are weak interactions which are playing the game here, but you need to understand that probably in all the LLPS, all of these are playing the game, but then what is the weightage of one another, that is actually dictating what you are going to see and what you are not going to see. Okay, so some of the experiment one is really interesting. It's called reentrant behavior. That means if you have, let's say, protein RNA kind of condensate, for some of the systems people have seen, if you start with very low salt concentration, you really do see those aggregates or those LLPS behavior. Then you go to intermediate salt concentration, nothing, it vanishes. Again you go to higher salt concentration, you again tend to see those. This is a very interesting reentrant. So it has something called, I mean, in temperature language is LCST and lower critical and higher critical temperature, something like that in terms of ionic strength and stuff like that. Interestingly, in one of the recent paper, they have designed a very simple peptide, charged peptide. It is proline arginine repeatedly for 25 mar, and they showed that this can actually self-aggregate to give you those kind of thing you see in LLPS at 2.7 molar of very high salt concentration of KCl or NaCl. So what in this study we are doing is very simple. What we are trying to see, we said okay, we will try to decouple some of the interaction going on in this kind of studies, and possibly we will be making some predictions to understand what's really going on here, why you are seeing these kind of things. First thing, we will be thinking about only interaction at the dimer level. Okay, we will try to form dimer, nothing else. At the salt concentration which is closer to them, and our design is very simple. It is Rx, Rx, R means arginine, X means it can go from glycine, alanine, proline or phenylalanine. We will also prepare other system where it will be EXX, E means here glutamate, okay, and then X can also vary. Okay, these are the type of system we are thinking about, and we are doing self dimerization, nothing else. How do we do that? Computationally, you can of course do that using something called metadynamics. It's a free energy simulations that we try to find out using our order parameter what is the free energy of the system. We have taken order parameter as the number of contacts the two dimer can form, and that we have biased in our system. We are varying this from 10 number of contact 10 to 100 and 10, and these are the some things we use some kind of cutoff to show that there will be some distance criteria where there will be a contact, and out of that if it goes beyond certain distance there will be no contact. Okay, so let's see what we find. First thing we found is that when your system has arginine, if you have glycine, these are two temperatures you are seeing, they don't show any propensity to dimerize at all. Only when you are increasing the level of hydrophobicity, that means going from alanine to proline, remember arginine is constant, it's a repeating things, arginine, proline, you find at high temperature actually you do see that higher contact forming stuff is getting stabilized slowly. Okay? But what is interesting, you don't see the same thing when you have glutamate sitting there. That means if you have glutamate, if these systems are not showing that propensity. All this is calculated at 2.8 molar NaCl solution. We have shown for arginine alanine system, we have just for purpose, we checked what happened for 1.4 molar. We have seen that propensity is actually decreasing. That means you do not find that much aggregation propensity what you have find for 2.8. But the message is that for arginine system you do find a propensity for dimerization, but not for the glutamic acid system. We said okay, let's see that what are the things we can calculate many things. Long story short, nothing is so different between the two systems. Number of ions, Rg, number of water also not very difficult, but what is really important here is that if you look at the residence time of water, you find there is a real difference. Around arginine, water doesn't like to stay too long, but around glutamate, they do like to stay really longer. I mean, literally longer, like 2 times or so. And that is actually reflected. These are, don't go to the numbers and plots here. You see the energy values, these are very pretty small, 1 to 1 minus 3, 3.3, but only interactions which is really important is actually glutamate and water interaction. That is actually strong interactions. No other interaction in terms of ion or water with all the side chains are important. I mean, important means number wise. It is the glutamate which actually preferred to interact more with water, and that is probably the reason it doesn't want to go to a state where it want to dimerize or go to a state where it can form aggregate. Okay? Is there anything else we can think about? Yes. You can see that these are actually side chain interactions. If you see side chain, these are for arginine system. You see more or less they are very similar at 2 temperatures. Only for proline at higher temperature you are finding little bit more interaction. That is probably because they are more hydrophobic interaction coming into picture and making the interaction more stable. But what is important is that if I want to now calculate self, like how arginine and arginine is interacting or how glutamate and glutamate is interacting, what you find, arginine arginine there is a favorable interactions when they are collapsed. But for glutamate you don't find such interactions. So it is a mixed effect. It is a solvation, water which is preventing not to go there. And other thing is that because arginine has this pi character, probably it has certain amount of indirect hydrophobicity coming into the picture, which people have talked about earlier, that is making the system with arginine more prone to dimerization, not the glutamate one. We went ahead and did something else also. For phenylalanine system, arginine phenyl, glutamate phenyl, we just did a simulation putting some of the 5 or 6 chains and tries to see what happened. We have seen the similar thing. For arginine system, we found they are all coming together and forming a single droplet kind of stuff, but not for again the glutamate system. And we characterize what is playing here. Of course, here cation pi is playing much more important interactions than any other, because you can see this arginine and this phenylalanine from 2 different chains they can form these cation pi interactions, which and solvation is also probably here hindering that effect not to come into the picture, because cation pi is not there. This is what I wanted to say for this very simple system, then how we can understand what is going on. Okay? And probably what I prediction was I am telling that if once make us things with glutamate, probably that is not going to go for a LLPS kind of transition, but we don't know, of course. This is kind of a signature coming from our work which we have done in 2023. Okay. Now, again, now forget about LLPS little bit, because this is something different I am trying to talk about. It is about protein folding again. So we know protein folding what it is. Amino acids, they can form chains, it can go to 3-dimensional structures, all this stuff we know. I will not bore you with this, you all know it very well. And you also know Anfinsen's experiment, famous experiment, that you denature the protein and fold it back, it always goes back to the same structure with this particular protein. And then it says that information contained in the protein sequence is sufficient to determine the protein structure. And thermodynamic hypothesis is also there. It says the native structure is the global minimum is the free energy surface. It talks about 2 things. One, sequence, if you know, that will give you a unique structure and thus function. And second is that this means it is not only telling that it will be in the global minima, it is telling probably the folding pathways are also defined, because it does not necessarily only define the minimum of the free energy potential, but the whole landscape from going from the unfolded state to the folded state. Okay. Now, the question is that this problem then has 2 parts. One is structure prediction. That means if I know the sequence, can I tell you what is the structure? That part people have done a lot of work. Let's first acknowledge that physics-based model has done fantastic job and everything. But then fortunately or unfortunately, I don't know how to say that, but in 2024, you know that Nobel Prize has gone to the AlphaFold, Google DeepMind AlphaFold solving this problem using AI. But don't go with their all the saying, this model only works when you use multiple sequence alignment and pair correlation kind of stuff very heavily, otherwise it doesn't work. So it is not necessarily a full black box. Okay, physics is already there. Okay, okay. First thing first. So there is structure prediction part is probably fine. What about the other parts? It is the how really the protein folds. So for that there is Leventhal paradox. We all know protein can astronomically high number of conformation possible. How it will select one? It will take too much of time. But then protein actually folds within biologically relevant time, which is pretty small. And this old work by Professor Joanjic, Atula Jabo and Biman Bakchi, they actually talked about this, and they said really interestingly that if you just think about a small bias, that means all these amino acids can have many different interactions. You just think that if they are in native state, different configuration one amino acid can have. In native state, this is 1 KT bias. or 2 kT bias compared to all the non-native states it can exist. That solves the puzzle. That says if you have that biased search, that time scale actually can come down to as small as 1 second. Okay? So it is, that means it is saying it is not a random search, it is possibly a biased search. How it is biased? They thought it is because of the native state which is being native interactions or native configuration is being stabilized compared to the all non-native states. Great. Other physics based model of understanding protein folding, one is Ising model. They are doing a binary kind of stuff, that means they are taking protein as folded or unfolded. But more sophisticated model is called spin glass model, where you can have both ferromagnetic and anti-ferromagnetic coupling within the spin system, and that gives you all kind of frustration you can have in your system. And you know that spin glass model can give you multiple local minimas and stuff like that. With some interesting variation of that model, this Brinjelson and Walliness is a famous paper of protein folding spin glass. They could show that if they think all the native state have some, all the native interaction have some slight stability than all the non-native interactions, where non-native interaction can be considered as a random energy with some Gaussian distribution and having some minima, having some average and distribution. I will come back to that. Then you can show that system can have phase transition from unfolded to the folded state, and there is something called frozen folded. I will come back, but don't worry about it. Folded and unfolded. Okay. But you understand something, that paradigm of classical structure function, sequence structure function has changed. Because now we have metamorphic proteins, we know the same sequence can give you 2 structures or maybe more structures, giving you more functions. If that was not enough, then you know that there are proteins which doesn't have any structure, so that means they are kind of IDPs. Okay? So things are now loose in terms of that particular Unfinsen's way of talking, that is becoming pretty loose. It can have, let's say, infinite number of structures. I don't know how many structures, but sometimes, not always, they can be fuzzy structure also. Sometimes they can actually bind to a target protein and give you a real 3-dimensional structure while functioning. All those IDPs I am talking about. Generally, they do not have any structure, but with target, they can go to a particular structure. It has been shown. Idea is, can I get a theory which can explain everything at once? Okay, this is just a try. We have done it for system where we said we have string of n amino acids. Each amino acid can have n number of native states, multiple, n can be vary, and 10 non-native states. That is fixed. That means I have some 10 non-native states, but it can have multiple native configurations, not one. Okay? And that is my n, because I want to do it general. Okay? But I will present first n equal to 2, okay? Because that is more tractable. So idea is very simple. I have string of n amino acids. Each amino acid can have 2 native interactions and 10 non-native interactions. That's the simple thing. If you think that way, okay, I will just go through, don't go to the equations, I will just, I have put it, but then I will just go through what I am trying to say in a simpler terms. So what I said, 10, you can have nu, which is equal to 10 our non-native configurations apart from n native configurations, and we are doing for n equal to 2 state, n equal to 2, that means it can have 2, let's say, for the time being. Now energy terms we have defined in simple. A particular amino acid at a particular position in which state? Is it in native state or is it in non-native interactions? And then Ising pairwise term. Is the 2 residue one after other, they are in native or one native, one non-native or 2 non-native, any kind is possible, right? That interactions terms are defined by some J term. But protein is 3-dimensional, so there can be tertiary interactions also. So it will also depend whether 2 tertiary interactions which are not close in the structure, sequence, but close in the 3-dimensional structure, whether they are native in nature or non-native in nature, depending on that there is an interaction Kij I can define. This is very simple thing. So then any configuration of protein can be written like this, very simple. It's the term which is residue level, this is pairwise, and this is all three-dimensional structural level. Yes, that's the model, very simple model. Now what I am asking, that let's say I have n0 number of states, n0 number of residue is in the native state, while my length is n. Some fraction is in the native state. That can give you many different kind of configurations, right? Energy, having different energies. They are generally not correlated if I think about random energy approximation. So it will be kind of a multiplication of all probability of having that energy of different energies, and I can multiply that. Now question is that this I can now, using the spin glass way of doing, Derrida and others have done, we said I will go to a stochastic approach. That means I say what this probability should look like. Because it is no correlation between one another, generally it will be Gaussian with some mean and some standard deviations. Okay? Fortunately, if we think something interesting here, then we can say what it is. Now, how I am doing it. So this is my mean and this will be my standard deviation. How I am doing it? Very simple. I said if they are in the native state, they have this certain energy, either epsilon 0 or j or k, depending on what kind of interaction I am looking at. If not, they are drawn from a distribution whose minima is certain little bit higher in energy with some distribution delta E. Okay? You can do a simple probability studies that if I have n0 number of states, how possible that next one will be my native or non-native. That way you can just do all these calculations to get very simple expression for this average of the distribution and the standard deviation of this distribution. Yeah. Which one? They are independent, yes, yes.
But why? So are you kind of probing equilibrium, or for equilibrium I can just write down the Boltzmann rate, right? In
Yeah,
terms of the
equilibrium you
different
can write down bold, but where I am going is that I am trying to find out what should be the distribution, and from there I will actually talk about something called microcanonical ensemble, like density of states approximation I will go. From there I will calculate the density of states from the probability distribution. And
So your ensemble is because I thought somehow it's a canonical. So
no, it is microcanonical.
you are considering a micro.
Yes.
Okay.
So now, once you have that, you can do that, okay? You can write down, just take it for now. From there, what you can define something called density of state. How many states are there at a particular energy? And that will have certain expression of this kind, okay? Forget about this for the time being. If you have this, if you have density of states, statistical mechanics tells me, if I take log and then I maximize that, that will be my entropy. As simple as that, because coming from microcanonical ensemble. So what I can say that from here, what I can define is actually the entropy of the system in terms of the order parameter, which is simple, is n by n0, n0 by n, or for a particular relative state it will n1 by n, right? Because you see, rho is the total fraction of native fraction, and rho 1 is a particular one, which is state 1. So 1 minus that will be simply your rho 2, the other state, how much will be the fraction. But now that is the beauty. Once I have that, I can use this expression to get back to the canonical ensemble and define my energy, entropy and free energy of the system. Once I have that, I can define the derivative of those. Once I have those derivatives at different value of L, that means this interaction differences, you see that how much is the native state is stable compared to the non-native state, I can have a phase diagram simply because I can solve this simultaneously. Yes.
What you were saying is that because it's microcanonical, so you have a strict limitation.
Yes, it has to land.
Those
Yes.
states.
Yes, exactly, nothing else. So, but you understand that, I mean, anyway, those, at least statistical mechanics says that even if you go to canonical ensemble, it is a delta function around that average value anyway, right? There is equivalence of different ensemble. Okay. So now, if you have this, you can solve that, and at different condition you can see the phase diagram, what should be your rho and rho 1, and that's what we'll do. But before that, just for the clarity, we actually derived for n, any n possible you can think, and we define all the derivatives and everything, and then you put n equal to 1, n equal to 2, n equal to 3, n equal to 4, 5, and get all the equations you want and solve for it simultaneously and get the phase diagram. We use simulated annealing, okay, for getting this all the phase diagram and everything. That means the maximization problem. Okay, now doesn't matter what I said earlier, just the result. What it's showing is that this is L minus L bar by T, means it is the tertiary secondary interaction taken together, L minus L bar. It is telling how much my native state energy between two residues, that means pairwise, how much it is stabilized compared to my non-native interactions. And this says how much is my width, because that is the entropy. So how much is my width? It is the average value wise, how much is the difference, and for non-native there will be distribution because it is random, how much is the width. That is the two parameter I am varying here. If you vary, what you will find really interesting. System initially in this region will be in the unfolded state, then it will cross a phase transition, first order phase transition, and it will go to folded region. How you can check that? You see rho low fraction of native contact. At the onset, it jumps to 0.8 or 0.9. Okay, rho is total number of fraction native contacts. So two things I am finding. One, all the systems, this is for 1 to 5, they are actually crossing this phase transition. In terms of total fraction of native contact, they are all doing this phase transition. But what is interesting, you see, as I increase my n, I am shrinking my folded region, right? Now think about, if your n is really high, what is going to happen? You will shrink really too much, then at a physiological temperature, which will be somewhere here, even if you have 10 number of native states possible, you will be in the unfolded state as far as the phase diagram is concerned. And that is your IDP, okay? That is your IDP. But when n is limited, then what happens? Like n 1, 2 or 3, you will see what happens. Look at the folded configuration for n equal to 1, 2, 3, 4, 5. Folded configuration means I am in the folded state, I am checking the health checkup of folded state. How? I know for total fraction they have all crossed the threshold, it's like folded. But I know for two state, there are two state possible. So I have to check each of the row. So the highest row value, I said row 1, next one I said row 2, okay? You see, for 1, 2 and 3, 1 is of course singles basin, it's a two basin, two possible state, three possible state, all of them goes to a high value of row 1. That means when they fold one of the particular structure, one of the three. However, when 4 and 5, first of all, folding region is shrinked, possibly they will not fold in normal region. Even if they fold, none of the structure will predominate. It will be a mixture of states. How do you see that? The highest row is decreasing. Here, the second lowest, second highest. What happens? Once it is folded, for row 2 and 3, there is a demixing. That means row 2 is decreasing, row 1 is increasing, and for 3 and 4 and 5, it is a mixing. That means it is the other roots are also increasing. That means there is a mixture of states in the folded state, if at all it goes to folded state. Yes.
I'm going to try and paraphrase you. Please say whether what I'm saying is right.
I think I am not doing good with time. Yes. Yeah.
Maybe I'll do it later.
No, just go ahead, please.
So, for every residue position, there are multiple conformations possible, and they are all at equilibrium.
Yes.
And then for the next one along, there are again multiple. The number may be different, but they are at equilibrium.
Yes, you are right.
And so on, right? And the IDP has not complete structural freedom,
Yes.
but a limited structural freedom where
Right.
at each position along the chain, you have any one of 1, 2, 3, 4,
Yes,
5.
yes. That's my point.
Right? And then when it all sort of precipitates into a single structure upon binding to the designated cellular partner,
I'll show that.
does it fall like one,
Yes,
with
yes.
cooperativity?
Yeah, I'll show you. I'll show you. That is the next thing. So this is fine, and all these plots are the same. I will not waste time. But what it is showing, at least in my setup, up to n equal to 3, it is all like singly folded. N equal to 2, what will happen? At free energy, we will have two basins. One fully one state, another fully two state. N equal to 3, we will have 3 basins. But each of the basin has one state. Right? Mixing comes when n is increasing beyond 3. Okay? So now, you see, this is this is just a comparison of 1 and 2. I will not waste time. But look at this. For n equal to 2, I can get actually the free energy diagram. I have, because for n equal to 3, it will be complicated. N equal to 2, you see in the unfolded region, you do see unfolded state is the stable, but then you have two basins which is unstable, equally stable two symmetric basin. Yeah, 3 is fine for me. At transition state, where you see the spike, you see all the 3 basins has very similar like stability. But in the folded state, you find two symmetrically stable basin folded. It's like metamorphic protein. Same will be for n equal to 3. Okay? Now this is interesting. What we said, for n equal to 4 model we have taken. Okay? So this red line you are seeing already shrink to 1 folded. This is unperturbed model. I just plot the n equal to 4 first order fluctuation. Then we should just do this simple thing. Out of the 4, designate one, the first one, and say there is a bias of 0.4 kBt or 0.5 kBt or 0.6 kBt. To say those bias in the native structure will come when it will bind to a partner. Nothing else. Just put a bias for one particular state out of the 4. Very little bias. Okay? That is only in the residual level, not in the pair level or anything. We have not done that. I mean, if we do pair level, it will be complicated. Okay?
For one more reason. In alpha helical structures,
Yes, you are right.
neighboring residues
Yes.
have interactions.
Yeah, I get your
At
point.
least I2, I plus 4, but in leadership
Yeah,
they don't.
you are right. So when I do that, what I find that you actually regain folded state. When you put that bias, you are actually going towards your folded state. Now folded line goes backward. That means you are regaining the folded state. That means there is a propensity that if the position of the sequence is here temperature wise, earlier it was unfolded region. Once you put this biasing, it becomes folded. Good. But how it is getting folded? Folding is exactly same as one state folder. That means you find only the state you biased that is becoming folded, all other demixed. That means what I am trying to say within this whole thing, that if I have a n native state description, there I can actually show that when n is finite but not 1, more than 1, there I can show that system can still exist in a demixing limit. That means it can go to metamorphic kind of states. But when your n increases slowly, your phase transition line is shrinking, making folding propensity closer to 0. That means at a physiological temperature, your unfolding probability is more, unfolded state probability. That means it will be unstructured. But moment you put bias on one state, phase diagram also shifts towards the right directions and you get folded to the actual folded state. That means which one you have biased. Okay? So we are trying to say that using this, we are kind of giving you a theory where you can actually take care of everything. Okay? Now, whether it is fine or not, that's up to judgment later on, but this is what I wanted to talk about. But I think I should talk about the first part. LLPS Devashish Saha, he has done this, and the second part is Rohan Moitra. He is currently working in my group, of course, doing on other stuff, and DST ACRB for funding, ISCS for some amount of computing, and thank you.
Thanks, Sridhar, for a wonderful talk.
Hi, very nice talk. So I was wondering for the re-entrant behavior. In the paper, I have seen that when increasing salt concentration, in the low concentration, the electrostatic interaction is the main driver. And when increasing the salt concentration, now the electrostatic interactions are hidden, and mostly the residual hydrophobic interactions play a role in the high salt phase.
It says interesting line. I have seen some paper. It
Yeah.
says hydrophobic and non-electrostatic.
Non-electrostatic,
So that means
exactly.
they really do not know what is happening. It's something like I would say hydrophobic and indirect interactions through ions and water rearrangement. That is what is happening probably. Okay. Anyway, yeah. Go ahead with
So
it.
I was wondering, have you looked into extremely hydrophobic amino acid residue and
We have looked for phenylalanine. There we found cation 5 is
Cation
the
5.
real factor, but we have shown for glycine, alanine and proline, and we show that for proline actually the experiment is also on proline. It is showing the propanol propensity. See, the point is that arginine is making sure they come together. Proline is making sure if you increase the temperature, hydrophobicity takes into account. Because
what
if you,
if there is valine isoleucine?
yeah, it will be there. I mean, I cannot check all that.
Right, right. Yeah, I was like thinking in terms of more hydrophobic.
I mean, I'm sure that those will be there effects, but we need to check. We cannot just say that it will be there.
Thank you.
Talking about the Leventhal paradox, most people have paid attention to how within finite time, through some pathway,
Yeah.
an accessible local energy minimum is attained, not necessarily demonstrably the global energy
Global
minimum.
energy, yes.
And all of it actually started with Anfinsen's demonstration of refolding. But there's also a very large part of the community which asks a very inconvenient question: where is the scope for refolding to occur in nature?
You are right.
So, an alternative explanation, right, which addresses the Leventhal paradox is co-translational folding on the ribosome.
On the ribosome, yes.
Where as the chain emerges from the ribosome, progressively longer stretches of chain developing through vectorial addition of amino acids during translation is actually creating at every stage of addition of amino acid a new conformation energy
Yes.
landscape.
And then that can trace back.
And if that goes to the thermodynamic minimum at that stage,
It will stay there.
from there, it may be a short hop to what happens when the
Yes.
next amino acid is added, and the next, and the next, and so on. So therefore, what refolding, when it occurs, and it doesn't occur, I study a bunch of proteins from hyperthermophiles which do not refold, right? So when it occurs with mesophile proteins and with single domain proteins,
Basically for small globular.
small globular proteins, single domain proteins,
Yes,
you know,
yes,
no disulfides, no prolines,
Sure,
and so on.
100%.
right? So where it occurs, it probably recapitulates co-translational. folding.
Co-transition. You're
That
right, you're
means-
right, yes.
That means nature has designed it for folding from the N-terminus to the C-terminus through gradual production. And when the whole chain is there and refolding and doesn't know what to do, the N-terminus starts off
Yes,
doing something autonomously.
autonomously.
And progressively, as you go towards the C-terminus, they pack against those preformed structures.
And it
so
is,
they-
it is
yeah.
also known that when you do co-transitional folding, it actually can sometimes go to something very different structure,
Yes,
which is not the native structure
Yes,
actually.
that's
And
right.
then,
in fact, for subtilisin and for alpha-lytic protease, it has been shown
yeah, I know.
that if you refold it, it goes to a totally different structure. So therefore, when an IDP forms a native structure after binding to its designated cellular partner,
Yes,
there is a possibility that the precipitation, I'm using the word precipitation here in
I got
terms
it.
of English, not in terms of science, right? When a state precipitates for each one of those
yes,
residues, it might actually also recapitulate co-translation
co-transition. No, you are right.
from one end of the chain.
Yes, yes.
So that sudden shooting up may be happening residue by
Residue by residue.
by residue.
Yeah, we haven't done the dynamics on the landscape yet. This is just the free energy part. We can do the dynamics on this. We have a plan to do it, but that is much more difficult, by the way. But we have a plan to do that. This will be complicated, but we will do that, the dynamics on this multi-dimensional landscape, how it works.
So with that, let's thank all the
Thank you.
speakers of the session.
Okay, I'll make a few announcements. So we have the poster session right now, and there's also again some coffee available.